Hello everyone, welcome to this online course. Today we are looking at microbial enzymes and their industrial applications. We'll be looking at different types of microbial enzymes as biological catalysts with their amazing industrial applications. My name is Bamidele Ojumosu. What are enzymes? Enzymes are biological molecules, biological substances produced by microorganisms, plants, and even humans. They're actually produced by every living organism, and these enzymes usually act as catalysts to bring about certain specific reactions within the cells. They help to bring out, they help to fast track reaction within the cells, and they are useful in every living cells. So enzymes are natural substances. They are produced within every living cells, both microorganisms, plants, animals. So the characteristics of enzyme, what are their characteristics? Enzymes have specific characteristics that distinguish them from every other substances that are produced within the living cells. One of the characteristics of enzyme is that they speed up chemical reactions, like I said. Some of these reactions are happening within the cells. They are also selective in their reactions. They're highly selective in their reactions within the cell. That is, they don't just act on anything, but they have a specific target. They have a specific reaction they catalyze. And these reactions is what the enzymes are actually synthesized for specifically. Another characteristic of enzyme is the fact that enzymes are highly efficient in their activities within the cell. They're highly efficient and they can be affected by environmental conditions. When we say environmental conditions, talking about pH, the acidity or the alkaline nature of the environment has a significant impact on the effect or reaction of enzymes within the living cells. Then they can also be affected by temperature. As a matter of fact, higher temperature can denature enzymes. So, if the temperature is the optimum temperature at which enzymes can act, the enzyme activity is not restricted. But the activity of an enzyme can be restricted by higher temperature, so they can be denatured. Then, enzymes can also be regulated within the cell. Their activities can be regulated within the cell. They usually have some, let me say, moderators, enzyme moderators. Some of these enzyme moderators allow enzymes to be produced or inhibited. Some enzymes are induced, some enzymes are produced constitutively. Induced means something can trigger it in the presence of a reaction. Constitutively means the enzymes are produced whether there is a reaction available or not. So the activities are usually regulated within the cells. And most enzymes are usually bigger, they are much larger than the substrate they act on. And only a small portion of the enzymes is usually directly involved in the catalysis. Like I said before, enzymes can actually enzyme can be denatured by heat or other chemical that may alter their activities. So this can also alter their dimensional structures. because some of these structures has its own benefits. Even though they are bigger, they are larger than the substrate they act upon, their structure permits them to accommodate some cofactors. These cofactors are also substrate specific. They can also act along with the enzymes. So when enzymes are denatured, it can alter their structures. So when these structures are altered, it usually affects the addition of cofactors and even affect the enzyme itself. And in such cases, it could be a reversible reaction or a non-reversible reaction. Enzymes can also contain sites that bind to cofactor, like I said. So these sites are actually needed for their catalysis. It allows them to have a bit more of a more specialized activity. So they have a binding site that allows some small molecules, which are often direct or indirect product of their substrate they are catalyzing. And these are usually, attached to their structure. So when they are denatured, some of these become disabled and become less functional. This is a diagram that shows an enzymes, an enzyme and a cofactor. The cofactor usually get attached to the enzyme and it has a binding spot. So this is the substrate. This is just an example of an activity that is being catalyzed by an enzyme and a coenzyme. So, sources of enzymes and their uses. I said previously at the beginning of the lecture that enzymes have origin from plants, microorganisms, and humans. So, microorganisms are one of the sources of enzymes, animals, and plants. So, there are various sources of this enzyme and they are used in the industry. And example of microbial enzymes include amylase, protease, cellulase, and so on. And we also have example of animal enzymes, pepsin, trypsin, and cellulase also, papain, and bromelain from plants. These enzymes, they all have their industrial uses. Cellulase is present in some plants, also in some microorganisms such as fungi. It usually helps to digest cellulose, which is a major component of plant walls. So, because of the fact that the component of the cell wall is cellulose, so the enzymes that help to digest it is cellulase and is present in both microorganism and plant. Now, uses of enzymes. Enzymes are used in various forms. We have them in recombinant technology, food preservative, biofuel, biofertilizers, bioremediation. In recombinant technology, briefly, involved inserting a desired gene into a suitable host organism, we then produce the desired enzyme. That's how enzyme is used in recombinant DNA technology. Example is Escherichia coli, which is usually found useful in biotechnology. For food preservation, some enzymes that possess antimicrobial properties can be used to extend shelf life of food products. And biofuel, certain enzymes are also used to break down carbohydrates into fermentable sugars for bioethanol productions. And also for biofertilizers and bioremediation, the enzymes that can enhance uptake of nutrients from the environment for plant development. And other enzymes that also help in degrading pollutants in water are useful in bioremediation, and they offer eco-friendly approach to environmental cleanup. These are examples of uses of enzymes generally. Enzymes are good alternative. Microbial enzymes are good alternative as a source of enzymes. They are good alternative. Enzymes that are produced by microorganisms represent alternative source of enzyme because they can be cultured in a large quantity. They also have a broad spectrum of activities. Microbial enzymes are also very stable than those of plants, and the major advantage of using microbial enzymes is the fact that microorganisms usually produce these enzymes, and there can be a bulk production. It has economic impact. It helps to get these enzymes in a large quantity, bulk production capacity. And the microorganism itself, they are easy to manipulate. Their cells can be manipulated. They can be the gene can be inserted to another organism to make it produce the enzymes, and they can also be used to drive some other enzymes that are available in nature. So microbial enzymes are the best alternative source for enzymes production uses, because like I said, they can be cultured in large quantity by fermentation, and because of the fact that microorganisms have diverse biochemical properties and biochemical pathways, they are susceptible to gene manipulations, and this enhance their uses industrially. Microbial enzymes are also cost effective, which means they can be produced relatively cheap due to efficient fermentation processes. It doesn't cost much. Enzymes can be produced in a large quantity via fermentation, so they are cost effective, and microorganisms can produce diverse enzymes with different functionalities. Even some of the enzymes can still be manipulated within a very short period of time. So microbial enzymes are they dominate industrial uses because of all these factors that I mentioned. And these are key information about microbial enzymes. That does not mean microbial enzyme does not have its own drawbacks. It has its own limitations as well. So let's look at the limitations of microbial enzymes. Drawbacks in microbial enzymes. One of the drawbacks is downstream processing of microbial enzyme. It needs specialized protocol because it has to undergo certain purification steps to remove impurities. These impurities are usually encountered during fermentation processes. We have cell debris, we have cell death, we have some other products that are released during microbial fermentation process which are not relevant for the target product, which is the enzyme. This has to be removed, and in removal of the enzyme impurities from the enzyme, it could be tedious. It could also require some specialized procedures in the downstream process. So this forms a lot of discouragement during the downstream process. This is one of the drawbacks of microbial enzymes. Then also, some microbial enzymes can trigger allergic reactions. The product itself can trigger allergic reactions, especially for those that are used for medical purposes. Some people naturally react to some of these products that are made from microbial enzymes. So these are some of the drawbacks, because some of the microbial enzymes are not compatible with the humans. Every other thing that is used in industry, basically, some of them are medical. They are for medical uses, and for medical uses, it has its own um, human angle application, apart from those that are used in the environment for cleanup and so on. So it can actually induce allergic reaction for the end users. So let's look at the next one, which is the plant enzymes. Benefit associated with plant enzymes. Plant enzymes has its own positive uses, even though it is not widely, patronized like microbial enzymes. Plant enzymes are generally recognized as safe, grasp, and they are actually considered safe for human consumption, and its application is widely accepted for human use. For medical use, plant enzymes do not usually induce allergic reaction. They work at all pH levels, especially the digestive enzymes in the human gut, and they also aid digestion, quick digestion. So plant enzymes are very good, though there are few that are used, but they're very good. They are also specific in terms of their uses. They have high substrate specificity. This makes them to be ideal for specific applications. However, plant enzymes also has its own drawbacks. Major drawbacks of plant enzyme is the fact that the seasonal availability. Enzyme yield from plants can vary depending on the season and growth condition. So when a plant is being used for a particular enzyme, the season at which the enzyme is obtained from the plant may be different from the one that will obtain the next season. For instance, during rainy season, plants give out different types of enzymes. At rainy season, at dry season, the same enzyme, but there could be some little bit of alterations, differences, because the plant is reacting based on the growth conditions. Perhaps there are no availability of water in the environment, or there are some intense humidity and so on and so forth. Then also lower yields. Production of enzyme from plant might be less efficient compared to micro because of lower yields. Enzyme may be potent, but it might not be enough. Then there's a lot of complexity in extraction of plant enzymes. It involves also specialized protocol for extraction purposes. So now let's look at classification of enzymes. Enzymes are divided into different classes, 7 main classes, according to the type of reaction they catalyze. So based on this, we have oxidoreductase, we have transferase, we have hydrolysis, we have lysis, and so on and so forth. In this lecture, and as a matter of importance, our focus will be mostly on oxidoreductase and transferases. Not to say that hydrolysis, lysis, isomeresis, ligase, and locases are not important. We will still come back to the oxidoreductases and transferase, but before then, lyses are those enzymes that catalyze the cleavage of carbon to carbon bonds, carbon to oxygen, carbon to nitrogen, and other bonds by elimination. The recommended name for them, for example, we have decarboxylase, aldolase, dehydratase, because they eliminate carbon dioxide and aldehyde and so on. For isomerase, they catalyze geometric or structural arrangement within a molecule. Their function is to catalyze structural or geometric rearrangement within molecule, while ligase, they catalyze the joining of two molecules together. They can join the DNA molecules together within a process. They can also catalyze joining of molecules that couple with hydrolysis of certain bond, for instance, pyrophosphate bonds in ATP or other nucleoside. Hydrolysis catalyzes the hydrolytic cleavage of certain bonds between carbon and oxygen, carbon, nitrogen, and some other bonds, and so on and so forth. But our focus, like I said, we are not going to really focus on these ones. We are going to focus more on high oxidoreductase and transferase. Why? Because we are looking at microbial applications of these, and that is where our focus is. So we are going to look at oxidoreductases now. Oxidoreductase comprise of a large group of enzymes that catalyze transfer of electrons from an electron donor to an electron acceptor molecule. So you can actually call them they catalyze redox reactions. The catalyze redox reactions, this is what they are known for. Example, we have dehydrogenase, we have oxidase, we have peroxidases. So types. Example is we have lactase, dehydrogenase. This one converts lactase to pyruvate, thereby transferring electrons to NAD. That is. We also have succinate dehydrogenase, which transfer electron from succinate to electron transport chain using the FAD as a cofactor. So oxidoreductase are important enzymes, and they are widely applied during microbial activities, and they are also used industrially. We also have another one, which is the cytochrome P450, which is a large family of oxidases involved in drug metabolism and detoxification, which involve use of various electron donors in the process. Transferase are class of enzymes that transfer functional group from one molecule to another. They catalyze the transfer of specific group such as methyl, acyl, amino, glycosyl, or phosphate from one substance to another. Their own function is to transfer the functional group from one molecule to another molecule. So we'll look at how this is achieved. They usually involve in many biological processes such as metabolism, DNA synthesis, and protein modification. When we say protein modification, they transfer the functional group from one protein to another protein, and once a functional group is added to another protein, that means that protein is already modified. For DNA synthesis also, they help to transfer some of these nucleosides into another one and make them to have a new sequence. So they are involved in various biological processes. So I said the transfer specific group, methyl amino glycoside or phosphate group. So they are very important. They are useful. Some of their uses are actually still evolving. Some of them are still being, under study. Unlike oxidoreductase that have an established application in the industry and in the medical settings. We'll be looking at them one after the other. Example of transferase enzymes, we have the kinase, transaminase, and methyltransferase. The kinase enzymes catalyze the transfer of phosphate group from ATP to a specific molecule such as glucose. So kinase is a very important enzyme that play a role in cellular signaling and energy metabolism. For transaminase, they also usually transfer from the word aminase. They transfer enzyme that they transfer the functional group from amino acid from amino group to another acid, so that's keto acid. So that is what they do. They are also important in biosynthesis of amino acids. Transaminases are important in the biosynthesis of amino acids. And we have the methyltransferase, which also transfer methyl group from another compound to specific compound or group, such as for instance, could transfer from adenosylmethionine to DNA or RNA. So methyltransferase play important role in epigenetic regulation and gene expression. They play important role in epigenetic regulation and gene expression. Epigenetic regulation of a gene is the process by which the activity of a particular gene is controlled by the structure of nearby chromatin. So now we want to look at the industrial application of enzymes catalysis. For pharmaceuticals, we have enzymes that are used in the production of antibiotics and other substances that are of pharmaceutical importance. Example is oxidase. We also have penicillin acylase. This helps in production of antibiotics that are useful for medical treatment. We also have enzymes that are useful in the food processing. Like I said in the beginning of the class, some of them are used for food preservative, and some of them are actually they help in improving the food qualities. For food processing, the application includes conversion of starch to glucose and so on, and also production of prebiotics, the between of food juice, and so on. We also have some of them that are also industrial use for detergents and making of paper, pulp. We have enzymes such as protease, lipase, amylase, cellulase. They act as a detergent. They remove stain. They are stain remover. They can also remove fat and oil. They also help in color retention in certain industrial products. They can be used as a detergent because of the fact that they have this ability to remove certain stains, rather than using chemical as a bleaching agents. Some of these enzymes can actually work as stain removers, and especially they also use in bleaching of lignin, and they also help in improvement of fiber properties. Some of these are actually applied in paper industries. They help to provide um, white paper for production of books and all that. We also have them in biofuel. Enzyme includes lipase, cellulase, and so on. So what they do is they help in producing bioethanol as a result of decomposition of lignocellulosic materials. So these enzymes are found applied in different, catalysis. They are found in application in different aspects. But industrial application of this enzyme itself is our main focus. How are they applied? What are the established products that they are being used for? Everything we have been talking about previously is about what surrounds them. But now we want to look at some of the established products that are being used, are being applied for, what these enzymes are being used for, that are everywhere. So first of all, we are going to look at oxidoreductase enzyme as biosensors. A biosensor is an analytical tool that comprises of a biological or biological derived sensing matter, which kind of have a close proximity to the physicochemical transducers. The chief function of such device is to produce a discrete or uninterrupted signal that is comparative to the concentration of the analyte. So enzyme-based chemical biosensors are based on biological recognition of this process. And in order to function, the enzyme must be accessible to catalyze a specific biochemical reaction, and it must be stable under the normal operating circumstances of the biosensor. So oxidoreductase enzymes are applied and used as a biosensor. So once the analyte is in tune, it gives a sensor, it gives a signal, and this is called biosensor. A sensor can be obtained in any form using some other ray of, maybe like can use some other form of inducers, can use electromagnetic waves and so on. But in this case, the sensor is biologically derived. So oxidoreductase is a very good enzyme for this purpose. It can also be used as diagnostic tools. The oxidoreductase enzymes are also very good in this aspect. The diagnostic tool is used in detecting disease condition due to the fact that they are substrate specific. It helps to carry out diagnosis, detect certain disease condition before they becomes pronounced in the cell. This is used in the medical settings. And how does this work? It depends on the fact that the enzymes are present in the cells, and depending on the um, severity of the disease, especially the state of the disease, it could lead to increase in the enzymes or decrease. For instance, if the disease is going to lead to tissue damage, in such condition, the enzyme that is specific to this disease or organ will be released into the blood circulation. And with a high augmented enzyme activity, the measurement of these corresponding enzymes in the plasma will give a signal that the disease condition is elevated. So this condition is usually a form of quantitative activity. So if the enzymes is low, then it could mean that the disease is not present, or it could also give a form of signal to mean some kind of organ failure. But if the enzyme is elevated, it's a signal that there is a damage or a disease condition in a particular cell, which is in this case a human being, because it will be detected in the blood. And the level of the enzymes in the blood is usually known. So elevated concentration of the enzymes in the blood can give a signal that this particular blood contains these particular enzymes, and in this case there is a sign of a disease condition, not an infection. In this case, we're talking about disease. For instance, cancer will also be tissue or organ defect. For example, we have the lactate dehydrogenase and sorbitol dehydrogenase. These two examples are enzymes that are released intracellularly. The lactate dehydrogenase is released during apoptosis or necrosis. These are major cell death in normal and disease settings. Now, for necrosis, which is a cell death, these enzymes are released, and there is an elevated concentration of this enzyme in the plasma. When this is quantified in a laboratory, it indicates a condition which could be maybe, for instance, cancer, because, for instance, sorbitol dehydrogenase are used for cancer prognosis, and the lactate dehydrogenase both are used for cancer prognosis. So anytime their concentration is elevated in the plasma, because they can actually permeate from the organ to the plasma. One time they have the level of this enzyme increasing, it shows that there is an indicator of cancer. Although this process must be accompanied by other tests, but it's like a diagnostic tool. which gives and give a kind of heads up during diagnosis. Let's look at other example of oxidoreductase uses in the industry. We have also in the generation of iodine. Lactase is one important oxidoreductase that is used in this process. The normal iodine, which are used for a lot of biomedical purposes, has its own drawbacks. So one potential application is lactase-based in situ generation of iodine. It helps in generating a more reactive iodine from iodide because of the redox reaction. Once these enzymes is in reaction with the iodide, it gives a more reactive elemental iodine, and this is used in the hospital and some other biomedical uses. Peroxidase is also a suitable replacement for lactase. It can be used for production of iodine, and like I said, this iodine can actually be used for several processes. We have other advantage of it in the biomedical uses. So the major application here is that these enzymes carry out a redox reaction and give a more stable and more desirable iodine from iodide, and is used for its normal biomedical processes such as sterilization and also for other antimicrobial uses. Other uses include air treatment. The current hair dyeing or weaving process that are used for cosmetic purposes are usually laced with chemicals and additives that are usually irritating to some users, or some of them can be very difficult to handle. So a lactase-based system may overcome some of these drawbacks because it can actually replace these chemicals by its own milder condition. So some of these hair treatments are now replaced by the enzymes, and it produces a satisfactory hair treatment better than those that are made from chemicals, which can actually irritate the users. Because of the fact that they carry out oxidation, transformation, and cross-linking, so they have been used in some of these satisfactory cosmetic hair use. In addition to providing an easy to handle hair care procedure, they also help in complementing cosmetic effects, which are usually achieved by conventional chemical methods. So lactase-based enzymes are actually used in this process for hair treatment. Another use of this include biodetoxification and biodecontamination of pollutants in the environment. Pollutants such as pesticides that are washed off from farms, xenobiotics, coal substances, and other industrial products that are derived from polycyclic and other halogenated hydrocarbons are usually found in the environment as recalcitrants. So these recalcitrants constitute hazards in the environment. If it's not taken care of, it could lead to some toxicity in the environment. So the use of these enzymes are important in removal of these toxic substances. Examples include chlorinated compounds such as tetrachloroethene. We also have polychlorinated biphenyl. They contaminate underground waters. Now, the oxidoreductases usually are powerful dehalogenating agents because of the fact that they are redox. Their action is redox reaction. They help to reduce certain compounds or oxidize it to make it either less active or reactive. So in this process, they help in removal of toxic substances from the environment and they help in decontamination, but biodecontamination. So in the use of this, there are other factors that are involved which complement their uses in the environment, but these are not usually well pronounced. We also have so many other oxidoreductase uses, but because of our time, we may not go into that. We only look at a few of them. And very quickly, we look at application of transferase. Transferase are commonly used in the biotechnology industry because of the fact that they transfer functional groups to other compounds. So they are used in the industry for the production of amino acids, antibiotics, and other compounds. I've explained previously the examples of transferase such as transaminase, which are used in the production of amino acids and so on, and they are relevant in the food industry and pharmaceutical industry. Recent advances in sequencing of the genome of environmental friendly relevant microorganisms and direct DNA sequencing of some soil microorganisms are revealing new metabolic pathways for some of these transferring for beneficial bioremediation purposes, but it has not really been established for now. Application of transferase in both medical and industrial purposes. For medical application of transferases, it is also used in diagnosis. Transferase play a vital role in medical diagnosis. It acts as biological markers for potential disease or damage to specific organ. Now, for transferases in healthy tissues, this enzyme usually maintain a certain level of activity. In this level, medical professionals can actually measure the activity level of specific transferases in the blood or other body fluids such as serum and so on. So if the level of the transferase enzyme show significantly increased activity in the bloodstream, that means there's a high level of this transferase in the bloodstream, it might indicate tissue damage, just like we talked about previously, previous application. It can indicate a tissue damage or inflammation near the organ where the enzyme is primarily located. That means the enzyme is usually present in the cell and they can permeate into the body fluids or the blood. So whenever there is increased activity or the concentration, it can also indicate that the organ is damaged or there's an inflammation near the position where the enzyme is located around the organ. So increased production of the enzyme by the organ affected can be as a response to disease. So the enzyme can react by being produced in higher concentration. However, conversely, if there's a decrease in activity of this enzyme, it could also suggest a kind of damage or malfunctioning where the enzyme is normally supposed to be abundant or at a normal level. When we say abundant does not mean it is high, but the normal abundant volume of the enzyme, if there's a decrease, it also indicates a kind of dysfunctional or damage. And if it is high, it is also an indicative of disease or damage. So for these particular cases, we have alanine transaminase, which is also known as ALT, and we have the aspartate transaminase, which are found in the blood and they can indicate liver damage. Their elevation can indicate liver damage or problem with the heart and the muscles. In genetic engineering, transferase also play a crucial role, such as in modification of DNA or RNA. They play some of this role by facilitating the transfer of functional group like nucleotide between molecules of DNA and RNA, especially in terms of DNA synthesis and so on. So we also have example of this in CRISPR-Cas9, which use a specific methyltransferase like enzyme called Cas9 for genome editing. So it is actually used in this process for genome editing, and these are applied in biotechnology. We also have the blunt end ligation and the labeling DNA molecules. For the blunt end, it's a restriction enzyme which are usually cut at specific sequences. Restriction enzymes are used in this process and they cut it as specific sequences, leaving some blunt end, and can be very sticky. But for blunt end ligation, where the two DNA fragments with blunt end need to be joined, so transferase usually come in a kind of DNA ligase, and it is used in bringing them together. Other enzymes like ligase also form in this process. So we also have transferase being used in modification of the DNA by adding some specific molecule attached to specific part of DNA, or some of them can actually act like labels or radioactive isotopes or adapters, which allow scientists to visualize or detect DNA of interest within a complex of mixture. It allows this process to go on and can usually identify this DNA. So it helps in labeling. So when it can transfer molecules to the DNA, which allows DNA to fluoresce. So we are going to end this class for now, and by the grace of God, we'll take it off from there. Thank you for listening.